Human biology operates at a staggering scale, from the complex systems of the body down to the microscopic operations of individual cells. It is driven by billions of molecular interactions happening every second. If we step back from the complex physical chemistry, we can view this system through an entirely different lens. At its core, the DNA driving these interactions is simply an information storage medium. The structure of this storage medium is precise. It is written entirely in a base 4 chemical alphabet consisting of adenine, cytosine, guanine, and thymine. The raw data load of a single human genome contains 3 billion of these character pairs. Bioinformatics is the specific discipline tasked with taking this massive physical polymer and translating it into a digital, computable format. The goal is to leverage machine learning models to accurately predict an individual's disease susceptibility, physical traits, or to identify targets for new drugs. But there is a technical limitation. Modern machine learning algorithms cannot natively process raw chemical structures, nor can they compute simple strings of text characters. To solve complex biological problems computationally, researchers must systematically abstract life source code into a machine-readable numerical matrix. To build this matrix, we first have to map the baseline biological data structure. The genotype, the genetic code, dictates the phenotype, which is the physical trait. DNA's operational role in this structure is essentially cold storage. It holds the raw instructions, but it does not execute any physical actions itself. To move information out of cold storage, the cell performs a copying process known as transcription. This animation illustrates the specific rule of transcription. The system duplicates the DNA sequence, but performs a find and replace operation, swapping every thymine character for uracil to create a new string called RNA. Once copied, the RNA enters the execution phase, known as translation, where this newly generated data is actively read by the cell. This table demonstrates how the biological system parses the data stream. It groups the sequence strictly in triplets, referred to as codons. The output of this parsing is a new alphabet consisting of 20 distinct amino acids, which act as the physical building blocks for proteins. We can view this mapping process explicitly as a programmatic hash map or dictionary lookup. The cell translates a 3-character input sequence into a single-character amino acid output. These resulting amino acid chains then physically fold into 3-dimensional structures to become proteins, the operational machines of the cell. Every physical trait we observe is the downstream output of this rigid, hard-coded biological execution pipeline. While this execution pipeline operates universally across humans, the sequences flowing through it vary between individuals. Statistically, human DNA is approximately 99.9% identical across all people. The data science challenge is that all predictive biological signal exists entirely within that remaining 0.1% of variance. To locate this signal, researchers align and compare thousands of genetic sequences from a population to pinpoint anomalies. This visualization identifies the specific anomaly we are looking for, a single nucleotide polymorphism, or SNP. It is a mutation of a single character at a specific location in the genome. A single SNP triggers a cascading algorithmic effect. By altering one base pair, the input to the 3-letter codon changes. Changing that codon input yields a different amino acid output, which modifies the final protein's 3-dimensional structure and its functional capability. That isolated structural change at the protein level directly drives phenotypic consequences, such as why one patient has brown eyes or higher susceptibility to disease. These isolated SNPs serve as the specific mathematical features, the independent variables, that must be extracted to build a predictive biological model. Before feeding these features into a machine learning model, we face a strict input requirement. Algorithms require purely numerical arrays, not character text strings. When looking at this sequence, the most common intuitive trap is attempting to assign ordinal numbers directly to the 4 letters, such as giving A a value of 1, C a 2, G a 3, and T a 4. Ordinal encoding fails because it falsely injects mathematical relationships into the algorithm. It incorrectly implies that T is greater than A, or that G is the average of C and T. In statistical reality, nucleotides are categorical variables. They possess discrete states, but they lack any inherent numerical order or magnitude. The data science solution for handling categorical sequences like this is a technique called one-hot encoding. This grid demonstrates the mechanical process of one-hot encoding. The single character string is expanded into 4 distinct isolated columns. The algorithm evaluates each position, placing a 1 in the active nucleotide column and zeros in the remaining 3. This effectively converts a text character into an independent true or false Boolean state. The final transformation required for inputting this data into a classifier model is flattening the 2-dimensional matrix into a continuous 1-dimensional array. This flattened Boolean array represents the exact milestone where physical, biological chemistry successfully transitions into a rigorous, machine-readable format. With the genetic sequence encoded, the final operational step is feeding these abstracted numerical arrays into a predictive classification algorithm. We can apply this to a specific, high-stakes genomic use case, searching for transcription factor binding sites. These are the specific regions of DNA that physically turn genes on or off. To train the model, thousands of encoded DNA sequences are paired with a known target variable indicating the presence or absence of a binding site, typically represented as a 1 or a 0. This diagram tracks the classification process operating at scale. The algorithm processes the arrays and actively sorts them based on the mathematical patterns that identified. Because the data has been abstracted, the model can detect imperceptible, highly complex nucleotide configurations that human researchers could never identify manually. The result is high predictive accuracy, achieved entirely by treating biological material strictly as a data engineering pipeline. This abstraction provides the foundation for personalized medicine, targeted drug discovery, and precision genomic engineering. Bioinformatics translates life's source code into a computable architecture, providing the framework to predict, manipulate, and engineer biological systems with mathematical precision.